Welcome to the second part of the PowerPoint on the cell. We are now going to be looking at some of the structures inside the cell. First, this is the nucleus. When you're looking at the nucleus, this entire unit, that is the nucleus itself. What makes up the nucleus is the nuclear envelope, which is the outer coating. It's similar to the phospholipid bilayer that exists around the cell. Inside, you have DNA. You also have it's a darker looking structure called the nucleolus. That structure makes something called ribosomes, which help make DNA, RNA, and other structures outside of the nucleus. Now, the nuclear envelope has pores. That's what each of these holes are. And that is where ribosomes and other structures move in and out of the nucleus. DNA cannot move out the nucleus. It's too big. So the pores only allow certain things out. Now we're looking again at the nucleus up here. And here's the nucleolus. And you can see the nuclear envelope. And you can see the pores. Now, what happens is the DNA in here, you can't see it right now. And when it's in the uncoiled, condensed structure, when it's going through cell division, it is called chromatin. And they're thinner strands of DNA. And I have pictures of that coming up. But we're going to be looking at DNA that's dividing, or cells are dividing, that called mitosis. That's in the next chapter. When we look at those chromosomes, they're visible underneath the microscope because they're preparing to divide. Here, they're not preparing to divide, so they're single strands, and they're called chromatin. Now, the DNA tells the cell what to do. And the way it does is the ribosomes travel outside through a pore, and then they get, here you can see how the pore is attached to the endoplasmic reticulum. Here are ribosomes dotting it. So that's called the rough endoplasmic reticulum. This picture is just showing you the surface of the nuclear envelope, and then these are the pores. Much closer picture of the nucleus. I put in some extra pictures just to help clarify what you're looking at. But this whole structure is the nucleus, there's the nuclear envelope, and there are the pores. DNA is inside the nucleus. And I had said before that it's called chromatin. It's a long fiber. And here you can see the DNA molecule, the double helix. And if you go back to chapter 3, we looked at that. There are nucleotides that hold it together, and there are 4 bases involved with DNA. Now what happens is it does coil up, and it coils up on proteins. And you can see that it gets thicker and thicker, and here are the single strands. That's a chromosome. You cannot see that because it's not condensed enough for you to see. Now, the number of DNA in a species or chromosomes depends on the species. We have 46. So here is the DNA, and you can see how it's coiling around the protein, and then it continues to coil on itself so that it can fit all of the DNA. You've got 46 chromosomes. They got to fit inside a tiny, tiny little nucleus. So for them to fit, they've got to condense somewhat. Okay, this picture, now it can start to get quite complex. The next chapter, not the next chapter, the 9, 10, and 11, we'll be looking at DNA and how it duplicates itself. And one of the structures involved is RNA. And there's a couple of subtypes of RNA. And you'll see a letter before it. This is messenger RNA. And what messenger RNA does is it reads the DNA. Because the DNA has the code, like computer code, to tell the organelles on the outside of the nucleus to tell them what to make. For example, you're a cell inside the pancreas. The pancreas make insulin. That's one of their hormones that they make. And it's critical for life. If our cells didn't make it, we would die. So the cells know what they need to do. They're told by the DNA. And it depends on what type of cell it is. Which again, the DNA is what has defined the cell long before you were even visible to your mom and dad. When the sperm and egg united, everything was determined at that point in time. So this is going to show you a little bit of where this mRNA goes. So here you have your DNA. We're inside the nucleus. And in turn, messenger RNA is copied from the DNA. And now we have a strand of messenger RNA. It goes outside through a pore into the cell, and it's here that it goes to the ribosome. The ribosome is the protein manufacturer of the cell. Messenger RNA got the code from the DNA. We'll say insulin. So messenger RNA has that code for insulin. It's got the computer code, but it's got to be brought out to the ribosomes so they can make it. They don't know what to make. The DNA is what's telling them what to make. Here we have the messenger RNA, and in turn, here's a ribosome, and it actually reads the messenger RNA and then creates the protein based on the amino acid. Ribosomes have been something we've been talking about already. Ribosomes are made up of two units. There's a large subunit and a small subunit, and they sit one on top of the other. Then there's a region in the middle for the messenger RNA to pass through. These make proteins, and you need the two subunits docked together to make the proteins. Now here on this slide you can see here's your ribosome, here's the messenger RNA passing through the two subunits, and it's being read by the ribosome. In turn, this protein is being made. Now, there's two types of endoplasmic reticulum. There's the rough ER and the smooth ER. The rough has the ribosomes dotting the surface, and they make the proteins. The smooth has no ribosomes on its surface. It is important for lipid manufacturing and detoxification of harmful substances. Here's our endoplasmic reticulum. Here's the nuclear envelope. See how they're attached so that the messenger RNA can get inside in order to communicate with the ribosomes. And here is a nicer picture of the smooth ER. Now, the cells have something called vesicles, which are literally little vessels or balls that pick things up, transporters. You could sort of think of them as your delivery trucks inside the cell. For example, say insulin was made. Now we've got to get it outside the cell. So it's going to carry the insulin, go to the edge of the cell, spit it out. And that is a term of active transport called exocytosis. So we can get a better picture here of how the proteins are being made. Here's the ribosome dotting the surface of the endoplasmic reticulum. Here's the protein or polypeptide. In turn, now you're going to have sugar or a sugar molecule come in and interact with the protein to become a glycoprotein that may be ready to be exported out or it may need to be worked on inside the cell. It's impossible to know from this picture because this happens most of the time. Now what's going to happen is a vesicle is going to be formed at the end of the endoplasmic reticulum. This is the glycoprotein, and it actually gets pinched off so that it can be transported elsewhere in the cell. There is another organelle that works with those vesicles, and it's called the Golgi complex. It sort of looks like flat pancakes on top of itself. What it does is it ships and sorts the proteins that were made in the endoplasmic reticulum. It's sort of like the UPS service where we send our mail. It's got to be sorted and sent to the correct address. Well, that's what this is going to do. The process here, this is the Golgi apparatus. You can see it's going to form another vesicle once it's ready, and it can bud off and go outside the cell. There are many different addresses that the Golgi apparatus can send something to. One of them is that it could create these lysosomes, which are very important inside the cell, and we're going to look at them in a little bit. Here's your Golgi apparatus. It forms vesicles just like the endoplasmic reticulum. So lysosomes are a sort of vesicle. Inside it, there are digestive enzymes. Now, these are in animal cells. Enzymes are proteins. these enzymes will break down larger molecule. Lysosomes have many, many different types of digestive functions. Some of them engulf nutrients, some of them eliminate things that were brought into the cell. They have many, many different jobs. Here are your lysosomes, and they're digesting food. Here it comes. Here's the food vacuole brought inside the cell. See how it's gone in? That's the plasma membrane. The two fuse, as you see here, and digestion occurs. And then that may be used inside the cell. Now, lysosomes can also break down materials that were brought in the cell. So here's your lysosome with the digestive enzymes, and this contains an old mitochondrion. You can see it right there. It's an organelle that we're going to talk about, but it's old. It needs to be replaced. So in order to replace, they have to eliminate the damaged one. So the lysosome fuses with the organelle and digests it and gets ready to expel it so that the body can eliminate it. This picture on the bottom is the rough ER. You can see the messenger RNA. And here is just a picture. This is really just showing you how the vesicle buds off. It's going to be pinched off here to become that. Now there are other vesicles that are called vacuoles. And there's one that's huge and very important, and one you're going to see underneath the microscope. And that is the plant vacuole. Vacuoles are storage organelles. They're very big. They're bigger than vesicles. And they may store water, waste products, chemicals, other cellular materials. In the plant cell, this takes up most of the space inside it. It helps make the cell more turgid or more able to stand up against gravity, because if there was no turgidity, it would collapse. Here's a picture. Here's your central vacuole. Here's chloroplasts. In the cells that you're going to be looking at, you're going to see in the Elodea leaf in particular, you will see the chloroplasts, and they actually stream around the central vacuole if you get it still alive. And I'll be explaining that in class, or you may have already had the class already by the time you listen to this. So here are transport vesicles. This is the rough ER. It's transporting something that it made. It takes it to the Golgi apparatus, which then sorts and determines which way it is to go. So here it creates another transport vesicle, and in this case it's taking it to the plasma membrane, and in turn it gets secreted out. Now vacuoles store many different cell products, and you can see here's another one. Here's your lysosome. There's a lot going on inside the cell. They're moving all over the place. So it makes sense that there's some kind of system inside the cell to help these vesicles know where to go. otherwise they'll be floating around inside the cytosol and not know where to go. So we're going to be looking at that in the next set of slides. Here you can see underneath the microscope the new vesicle forming. Here's the Golgi apparatus. Okay, here's your Golgi apparatus, this whole green thing. And here's a vesicle. And it's coming to the Golgi apparatus in order to be sorted and shipped. But I want you to see this yellow. That's going to be very important. And I indicated before in the last slide that how do they travel? How do they know where to go? We're going to be looking at that in the next set of slides.